All right, excellent. Okay, well, it's a pleasure to have Prabesh Kanel speak today. He, most of you, I think most of you know Prabesh. He's the co-director of our imaging core in the Udall Center, and is an assistant professor in the Department of Radiology, and his background is in computer science and image analysis, particularly MR work. But he's going to talk today about voxel-based principal component analysis of brain PET images. Okay, Prabesh, thanks very much.
Thank you for a very nice introduction, Roger. Yeah, so I'll be talking about like voxel-based principal component analysis. It's a multivariate method. It's a little bit different than like the univariate methods that we generally see and use, which is the SPM-based methods where like each voxels are treated as a single data point, and like, you know, you do a like statistical analysis on those voxels, rather than like in principal component where it's a multivariate analysis where it's a little bit different because it's not only like look at the covariant pattern within the data, but also do a correlation analysis. So wait. Okay, yeah, so the first thing like we'll be in this one, we'll be looking at like number of different imaging type and like how we applied and like different pre-processing methods that we apply on those imaging to come to a point where we actually do a principal component analysis, and like how we can use like the principal component that we get from applying a PCA into like the kind of analysis we want to do. So the first one we'll be looking at is like the amyloid image, like of 165 subjects. It's a parametric image created using Logan plot methods. Here we use like the cerebellum gray matter as a reference region, and we take out the white matter from those images because, like, as we know, like amyloid binding at a white matter is very, very high. So we are more of interested in looking at like the amyloid binding patterns within the gray matter regions for different groups. So we have like 44 subjects, like 80 Alzheimer's disease subjects where like all of them are amyloid positive. I'll come to in next slide where what does amyloid positive means and amyloid negative means. We have like 38 with DLB, dementia of Lewy bodies, where 13 out of like 38 are amyloid positive. And then we have like 50 participant like images of Parkinson's disease. All of them are amyloid negative. And similarly, we have like 33 controls and all of them are amyloid negative in nature. So the first steps that we do is that like we take a T1 MRI image from each participant, and then we segment them into gray matter and gray matter with subcortical regions that include the subcortical regions. So this is done using like the free surfer pipeline. And what we do is like we take the gray matter marks and then like calculate the mean distribution volume ratio from a registered PET image. And then like if the value of like DVR is greater than like 1.17 mean DVR value within the cortical region alone, that doesn't include any of the subcortical region, then we put them as an amyloid positive. One quick question: do you guys see like the video of like the video, or you only see the slides? Because like what I'm seeing is like one side is blocked on my side, so or you see the entire slide?
What I see is a bar that says image pre-processing, and then I see an array of PET images. Is that what you're seeing, or?
Yeah, wait, let me try to, okay, yeah, I try to minimize it, but like I wasn't able to see the PIB positive on the right side and mean DVR, but now I can see more of the thing. Okay, yeah. So anyway, and then what we do is like we actually take like the gray matter marks with subcortical regions and remove all the white matter. reasons from the free surfer segmentation, and then like we use the pet image, register pet image, and we maxed out all the white matter reasons from the pet image, and then we put them in MNI space because that allows us to do like voxels to voxel comparison between subjects. So and because these are like all parametric images, so we can actually do comparison between them. So the steps involved like for PCA that we implement is like the first thing that we do is like we remove all the voxels that have like 0 or negative values. And one of the reasons why we do that is like in multivariate analysis, one of the limitation is that like it takes a long time for us to do the calculation because it actually calculates all the voxels and like within subjects and between subjects too. So removing those values means like we are only concentrated on like the voxels that we are interested in, which is like the gray matter with subcortical regions. Then what we do with the remaining data set is like we log transform the data so that each voxels, like we apply log. And then what we do is like we center each individual data. So that means what we do is like for each individual voxels within a subject, we subtract them with like average log values. So all the individuals have a mean of 0. That means like that helps us to prevent like a single subject image having too much influence on the result. Similarly, what we do is like we try to like center each voxels, and this is done by subtracting the average log values among all the individual voxels and have the mean across individual for one particular voxels to be 0. And this also prevents us each individual voxel from having too much influence over the result. So once we like center like the individual data and center individual voxels, we apply principal component. After applying principal component, like so the first in the first case, we only took like 80 versus DLB as an example. And for that one, like we have like total of 82 subjects, combined of 82 subjects. And when we apply a principal component on like principal component, we get like principal component analysis on those data, we get like 81 principal component. So each principal component tells us like how much variance that it can explain within the data. So if you can see that like the first principal component can explain almost like 11% of the variance within the data. And if we take all 81 principal component, it can explain all the variance within the data. So on this example, we are showing like the first 3 principal component shown like with a in a fire color, like with the negative weight as close to black, and then like the positive weight close to bright yellow, like yellow or white. So yeah, then what we do is like we calculate like subject scaling factor, or it's also called like prediction factors. So basically what we do is like from each subject's individual subject's pet image in MNI space, we do a dot product with like individual principal component to get a values, which is a scalar value. And so we get for all the principal component, like 81 principal component for each subject, we'll have like 81 different subject scaling factor. So if we look at like, this is one of the like done in like box, like in a box plot, sorry, bar graph. Here we can see that like the subject scaling factor predictor score for like for principal component 1, principal component 2, and like up to 6, and like where the x-axis is the principal component, sorry, x-axis the subject number, and like the y-axis is a predictor scores or subject scaling factors. So for example, now when you look at it, like you can see like, especially if we focused on principal component 2 and 3, it seems like, you know, the one on the left side of like orange line, it's like subject scaling factors for subjects with Alzheimer's disease, whereas like the one on the right side is like subject scaling factors for the dementia of Lewy bodies. So if you look at it, like most of the subject scaling factors on like the principal component 2 on like it looks like it's positive in nature, whereas like the subject scaling factors for like DLB is negative, it's below 0. Similarly, if we look at like principal component 3, we can see that like majority of the subject scaling factor is positive for AD and like negative for DLB. Whereas like for principal component 4, it's actually opposite. So like the subject scaling factors is negative and positive for DLB. So what we do after that is like we try to select the principal component that actually tells us like that can separate like the predict AD versus DLB basically. So the way we do it is like we use linear regression methods on subject of principal components and choose the one with like the lowest AIC. So when we apply that, like when we do linear regression, what we found out is like the principal component 2, 3, 4, 5, 7, 8, 10, the combination of those actually gives us the lowest AIC score. So, I mean, which is not very surprising. If we look at like the previous slide and like 2, 3, and 4, we see that like those subject scaling factors actually separate the group pretty well. And so this is like once we combine these all principal components from the one that we get from like the AIC, we can see that like the weight are adjusted based on like the combination of the principal component, and now we can see that it actually ranges from like 0.001 to 0.0121. So now when we are selecting like the individual voxels, like because when it comes to principal component, so the principal component itself actually gives us the direction of greatest variability in the data set, but like and it uses all the voxels. So just taking like individual voxels weight on the individual voxels might based on its variance and correlation with other voxels. So what it means is like, basically what like when we are looking versus AD and DLB, and we are taking like combination of principal component, when we are looking at a voxels in principal component 1 to principal component another, like the weight for that voxels might be positive on one of the principal component, where it might be negative on like the another principal component. So, that doesn't mean that like whether that voxels is actually valuable to distinguish between AD or not. So selecting that voxels is a key in like telling us whether that voxels is important or not. So one way what we do is like we use bootstrapping methods and compute a Z map for each like voxels to find out whether those voxels are important like individual voxels. So when we run like the bootstrap, like the general rule is like if the Z value is like less than 2, then we don't consider it as good. But like if it's greater than 3, we consider it as like I mean, like the absolute value is greater than 3, then we consider it as a good values, but absolute value is less than 2, then we don't consider it as good. And we also have to be careful is that like, we might find a covariance pattern that explains the data well, and given good behavior correlation with lousy G values. So this doesn't mean that like the analysis failed. It just means that like the pattern that we have, every voxel participant in it to a small degree, so we have to be careful when we actually look at the data. So on our example of like AD versus DLB, the amyloid data. This is like this is what we found out. Like, and we are only showing, if you look at like the G value, we are only looking at like G value that is greater than 2, because like at least from our understanding, like G value less than 2 is not significant. So we don't usually take those values. And what we found out is that like in case of AD, the amyloid bindings are like more seen in like bilateral insula, anterior and mid cingulum, frontal lobe, parietal and post-central, whereas in DLB, it's more in like inferior, mid, and superior occipital lobe, thalamus proper, fusiform, and like calcarine. And we can also see that in DLB, the amyloid presence, like at least like covariance pattern, are higher in medial temporal lobe. But like if you look at the medial temporal lobe as a whole, like the volume, like the number of voxels in AD is much more less than DLB, which is expected due to atrophy. Similarly, if you look at like the distribution volume ratio, mean distribution volume ratio within that region, AD actually has like the higher distribution ratio compared to DLB. Similarly, like if we look at like the data for AD versus Parkinson's, what we see is like, bilateral insula for like where we see like amyloid presence higher, anterior mid posterior cingulum, frontal lobe, that's where we see like higher amyloid presence, whereas in PD, it's thalamus proper, fusiform, caudate. Those are the reasons where we see like more of amyloid presence. And similar case with over here, it's like the AD has like higher mean DVR in medial temporal lobe, whereas like where it compares to volume, actually it's lower. And in both like AD versus DLB, PD, AD versus PD, and AD versus normal controls, what we can see is like, if we just look at the volume within there, we can actually separate the two groups pretty easily. But what we are looking at it over here is more of like the presence of like amyloid, how they are distributed within each group. Similarly, this is the one for like AD versus normal control. We see like the similar pattern, like in all three groups, it's bilateral insular, frontal lobe, anterior mid posterior cingulum, and supplementary motor areas. Now, and in all of this case, the R square is like pretty high. And now in case of PD, DLB versus PD, we see the bilateral angular calcarine mid and post cingulum for cunus, precunus, and supplementary motor area for DLB. That's where the presence of amyloid is much more higher, whereas like we see like thalamus proper, insular, and caudate, where we see the presence of amyloid higher in those areas. At least in this one, what we can see is like we cannot separate DLB versus PD just looking at mean distribution ratio on major temporal lobe, because it's like pretty similar. Whereas like the DLB versus PD, if we just look at the volume, like DLB has like much more atrophy compared to Parkinson's. And we can actually separate DLB versus PD in these two. Similarly, if we look at like the DLB with the normal controls, we see bilateral angular calcarine. I mean, similarly with frontal lobe for DLB, and supplementary motor area and mid superior occipital lobe for where we see like the presence of amyloid when we compare to normal controls. And it's the same in this one too. It's like, if you look at it, we cannot separate like just looking at the mean DVR values. But at least like with the volume, we can have like some like, with a p-value of 0.004, we can separate the two group, but it's like, I'm not sure if it will pass. Now, and if we look at like the normal controls and PD Parkinson's, what we can see is like the presence of like the DVR doesn't exactly uh, in medial temporal lobe doesn't separate the two group. And similarly, if we just look at the volume of a medial temporal lobe, we cannot separate PD versus normal controls. But like at least what we can see that the presence of like amyloid are like present in like thalamus, putamen, palladium, and hippocampus in PD cases. Yeah, that's like for that's basically what we did with like the amyloid images and like looking at the different groups within the amyloid. Just taking like the amyloid images of like the DLB, AD, PD, and normal controls. Now we are also looking at like the another one where it's more for, we are looking at the serotonin DSP PET scan, and these are also parametric images created using Logan plot method. Here the reference regions is like infero-posterior cerebellum. We do partial volume correction on this data in MNI space, whereas if you remember the previous data, we didn't do partial volume corrections. We actually took the segmentation from like free surfer. And what we did on that one is more of like erosion, so that like the boundary voxels are eroded out. And in this data set, we have like 50 subjects with like Parkinson's disease, and then like 20 subjects with normal controls. Here, for segmentation methods, we actually use SPM-based segmentation. Here we can see like the blue area, like the T1 images are segmented into gray matter, white matter, and like cerebellar spinal fluid. The blue one is for the gray matter, the green is white matter, and then like red is cerebellum spinal fluid. Then we use like this segmentation and apply like the Muller-Gardner best partial volume correction methods on our like DSP registered DSP PET image to get like partial volume corrected PET image, serotonin PET image, and then we apply like we put them in MNI space using the same methods as previously done. And if you look at the predictor scores for this one, what we can see that like the principal component one, it seems like in both the group, like it's all negative, with like two subject actually having like much more influence on like the on the principal component one, and like principal component 3, like majority on normal control. Like the one on the left side of orange is normal controls, where we see like more of like negative predictor score and like higher predictor score on like for the PD. And so we do the same methods of applying like AIC methods, and like, and then we take a combination of principal component, and what we get a combination of principal component to get a G values by using a bootstrap methods, and we calculate the G values for the DASP PET image. Like, and here what we see is like the in PD, the presence of like the binding is higher in cerebellum, frontal lobe, and supplementary motor areas, whereas like in normal controls, like in PD, it's lower compared to like normal controls in like cuneus, precuneus, and occipital and angular areas. So this is like another example of one where we do where we did a PCA on like acetylcholine images, where we actually look at like the DLB and Alzheimer's. Like we had like 5 DLB subjects and like 6 Alzheimer's subjects. Both were with a vesicular acetylcholine transporter PET images. We did partial volume correction, and like 80 in fire color, DLB in blue to green color. What we see is like the acetylcholine loss in like as shown by the fire color for 80, it's mainly in like inferior mid superior temporal parahippocampal gyrus metathalamus, whereas like DLB it's a mid cingulum thalamus proper striatum and cerebellum areas. That's where we see uh, yeah, I'm pretty close at finishing anyway. So lastly, it's uh, this is like one of the example where the first three like principal component, we did it over here on our own data, like with different tracer. But like in those cases, what we mainly did is like we try to look at like how we can get a pattern that actually distinguish between the two groups. But like the principal component can also be like the methods, like this multivariant principal component based methods can also be used for to look at like the special covariant pipeline and try to do a analysis with like some of the test batteries. So this is like an example that I took from like this paper published in Brain Communication. And like their methods at the beginning is pretty similar to what we do, is that like they take all the images, like, and then they do like this is for the dementia of Lewy body subjects. And what they did is like they did a principal component analysis on those images, and then like we get like different principal components. And if you look at it, like they also calculate like the subject scaling factor for each and every principal component analysis. And then like you do a regression, linear regression or regression with like outcome measures to find like the minimum AIC values. And then like you use the reduced subset of like the principal component to come with a spatial covariance pattern the way we did to separate between the two groups. But in this one, what they did is like they were trying to look at like the M1/M2, like cholinergic M1/M2 like pattern in DLB patient, where these patients were treated with donepezil, and then like see like how uh, it changes. And they were able to correlate with some of the scores. So on the first one, like they did it with like the cognitive. If you look at the top, like A on the figure. So basically what they did is like they took an image at baseline, and then they calculate like MMSE scores, like NPI total scores. They also calculate like the hallucinations. And basically what they did is like after giving a medication, they came back after 12 weeks, and they uh, did the, like they had another clinical round, and like did the same test battery. And based on that one, like they calculate like percentage of like MMSE change, and then like percentage of NPI change, and like hallucination. And on that one, like when they did uh, with the principal component analysis the same way, when they look at the scores, what they found out, like there was an increase in temporal pole for like in cognitive. The binding pattern was like they found out like temporal pole increase in temporal pole, super mid orbital frontal gyrus, fusiform, and anterior cingulate. Whereas like there was decrease in like the middle temporal lobe, basal forebrain. Similarly, when they did it with neuropsychiatrics, like the imaging was done only in like the baseline. So the pattern actually that they found out was like there was increase in like for the neuropsychiatric, which is like the total NPI score. Using the total NPI score, they found out like there was an increase in fusiform, inferior mid superior temporal gyrus, and striatum, whereas there was a decrease in insular, basal forebrain, posterior cingulate area. Similarly, when they looked into the hallucinatory, where they look at like NPI hallucination score, they found out that like there was an increase in fusiform, inferior temporal, and caudate, whereas there was decrease in anterior and mid-cingulate medial frontal. That was the pattern that what they found out, and that's from like Coloby paper, the same paper as previously. Yeah, and yeah. So finally, what I would say, like the advantage of PCA based multivariate data analysis, it's like we can actually evaluate correlation and covariance of activation across brain region rather than looking at it as like voxel by voxel basis. So this result, like this pattern that we are finding, can be interpreted as a signature of neural network. Similarly, the univariate methods, which is like the SPM-based voxel-based morphometry method, uses very stringent and often conservative correlation for voxel-wise multiple comparison. So even though we see like the pattern forming by voxel basis, it's sometimes hard to pass through those multiple comparison because of like the number of voxels involved. And then similarly, it also provides information about like mean differences and correlation with behavior, and similar to what we get from like the univariate approach, but like it has a greater statistical power and better reproducibility. Yes. And finally, I would like to thank to all the collaborators, like especially Dr. Nicholas Bonin, Kurt Fry, Dr. Rosa Robin, Dr. Martin Muller, and Dr. Robert Kepe. I would like to special thanks to the staff at the Functional Neuroimaging Cognitive and Mobility Laboratory, and most special thanks to our research volunteers, without whom our work would not be possible. We need data, more data to. Thank you.
So, Besh, so I think probably the using the pattern analysis. I mean, this is a little, particularly for looking at individual factors. I mean, this is what David Heidelberg did with the analysis of FDG PET. Yeah, but so what are you thinking about for using this type of PCA for any specific predictors for clinical features at this point? I mean, what's coming up?
Yeah, I mean, like what David Heidelberg, like the paper that I've seen, what he has done is like he actually looks at like individual principal component and like try to do a correlation with like some of the like scores from some of the test battery, like with the MOCA, and then like come up with like principal component that and then name those principal component as a for motor or like, so that's how he does that. But like one of the thing like I was thinking about like using this methods is like the way the last paper did, where they were actually looking at like whether there is a pattern that explains like the change in like the MMSE scores from like from the baseline to 12 weeks. The imaging was done at baseline, but they were able to see like the reasons that had a high like increase or decrease. So I think like we can use do similar things because we have like a lot of data on amyloid for Parkinson's that were taken at the baseline, and then like and many of the people came back after like 3 years, but we didn't do 2 or 3 years. I need to double check on that one, but many came 2 years and some came 3 years. And what we can do is like we can look at like how those score changes on those test battery and try to find out like the pattern on like our data, whether it's like amyloid, or we can also look at into the cholinergic
So I guess
data.
So whether or not there are PCAs that will predict their clinical progression, correct?
Yes.
Yeah.
Yes.
Yeah,
Like the combination of PCA or
Yeah, I mean, that's what we'd like to try with the FEOBD baseline data, I think so.
Yes.
To try doing that for sure. Yeah, absolutely. Any other questions for Prabesh? I mean, these are potentially powerful methods that I think people should think about using. So. Okay.
Yeah, I think like one of the problem with this, like the more I try to understand this method is like what I found out is like the main reason people are not using this method, it's more due to the complexity and like the calculation and like the time it takes for us to do it in the but like what I like after working on it, at least my understanding, it's very straightforward, it's easy, and it's explained. Yeah, so
Okay.
yeah, definitely a method to.
Yeah. So, but my understanding, these are basically in many ways linear methods, correct?
Yes, the one like
Yeah.
where we actually look at like the combination of principal
Yeah.
component, like that's where it's linear, but like we can always like I mean, people have used like lasso regression too at that stage to come out with like the number of different principal component, and lasso methods to find out like choose the number of voxels too, so that like it's much more stricter. Yeah.
Okay. Interesting. Okay. All right. Very good. Okay. Any other questions for Prabesh? Okay. All right. Very good. Thanks very much then. So.
Okay.